Alright, so this is video 3. We're doing 3 different tests. The MRVP, which consists of 2, the MR or methyl red test, and the Volk-Proskauer test or VP test. And then the third one is called the citrate media. Okay, so to do the methyl red test, the purpose of it is to determine if glucose is fermented in mixed acids. And then your principle should include MRVP media is used. I'm going to explain that in a minute. The indicator methyl red is added after the bacteria grow. And if glucose is fermented, then when you add this reagent, you'll get a red color, and that means it's positive for methyl red. So what you do before you do the methyl red test, right, is you inoculate what's called the MRVP media. You use this media to do 2 tests. Okay, so the bacteria grow in this media, which contains glucose. After they grow, then you split the media into 2, and half of the media is used for the MR test, and you use the other half of the media to do the VP test. So you have to split it. That's what we'd have done in lab. So if I had the MRVP tube here, I'd inoculated it, the bacteria grow. In the following lab, we split the tubes in 2. Right, the same media is used to do the MR test and the VP test. So to do the MR test, which is I'm explaining here, if the bacteria converted the glucose, if they fermented it, and it converted it to what's called mixed acids, there are many different types of mixed acids. To simplify it, we refer to them as mixed acids, more than one. That lowers the pH. When you add the methyl red, that's the reagent or the indicator. If that remains red after you've added it, it's a positive test. If you add the methyl red and the media doesn't change color, then it's negative. They didn't ferment glucose. Or there's another scenario that could happen. What could have happened is they convert the mixed acids and turn them into neutral products. So they just kind of go a step further. Some bacteria can do it, some bacteria can't. So if they convert the mixed acids to neutral products, the methyl red test will be negative. Okay, so that's what you do for the second tube. You would use the same media to do the VP test, Volk-Proskauer. So you use the same media that you used for the MR test. Now you want to know, the question is, or the purpose, did they produce neutral end products from the mixed acids, right? That means the MR test is negative, they converted them, did they make neutral end products? So to answer that question, right, your principle should include VP media used, the reagents to determine this test. They're added just like the methyl red, but you don't add them to the methyl red test, you do it to the VP test. You add VPA and VPB to detect acetone. That's one of the neutral end products that's produced. So if that's present, then you get a red color and the VP test is positive. Volk-Proskauer. If it doesn't turn red after you add the VPA and VPB, then it's negative, and they didn't produce the neutral end products. So if they don't ferment glucose, this test will be negative and this test will be negative. If they fermented glucose just to mixed acids, then only this test is positive, this test is negative. If they converted the mixed acids to neutral products in this test, then they fermented glucose and they eventually became neutral end products, and this test is positive, right? So EA is positive. There are other bacteria that are positive for it, but you know, E. coli is usually positive for methyl red. Enterobacter aerogenes is usually positive for the proscour. So like all these biochemical tests are used to identify bacteria. You know, it would take, you know, it'd take probably 3 or 4, maybe even 5 or 6 tests to actually narrow it down to which bacteria you have. It just depends. Sometimes it's a slam dunk and sometimes it's not. E. coli is always a green metallic sheen on EMB, right? So that would identify your unknown for sure, without any other tests. Okay, so, right, your purpose is here, your principle, using complete sentences. These are real simple principles. The MRVP media is used for this test. Have the bacteria grow. You split the media so you have one for the VP, one for the methyl red. You add VPA, VPB to conduct the methyl red test. If it turns red, then you should mention acetylene as a neutral end product that will, if it's present, will turn red and make this, then you would consider this test positive. So the other test for this week is the citrate. This one's a pretty straightforward media. I got an empty tube here. I didn't inoculate. We had some problems with the bacteria, but it's a slant media, and you would inoculate. You take your loop, and you would inoculate the surface of that slant. I don't know if you can see it all that well. And then you actually stab this to the bottom too. So it's a stab and sweep, I call it. Then you allow the bacteria to grow. So in the citrate tube, the purpose of it is, can bacteria use citrate as the sole source of carbon, is one way of putting it. They're basically fermenting the citrate, right? There's no other carbon source in the media form, right? So that's the only, say, carbohydrate. So in your principle, then you would say, if the bacteria have citrate permease, then they can utilize the citrate. The indicator bromthymol blue appears green right before the bacteria grow. If they grow on this media, then you get a blue color. It's almost like the color of this cap. Really nice blue. That means citrate was fermented. They're positive for this test. So this is a really straightforward. There's no inhibitor in this media. There's no inhibitor in the MRVP media either. So you know it's not. You don't have to include them, but you should include, right? Remaining green is negative, and a blue color indicates citrate was utilized. So those are pretty 3 straightforward media for lab number 3, or lab video 3.